Good morning, everyone. So, in this set of lecture, we will be talking about parasympathetic nervous system. In the previous lectures, we gave you a brief introduction about autonomic nervous system, and today we will be discussing bit in depth about different classifications of autonomic nervous system, and one of them is parasympathetic nervous system, or it is also known as cholinergic nervous system, right? So, what is basically parasympathetic nervous system or cholinergic nervous system? So, whenever you are in a very relaxed condition, okay, whenever you are relaxed, suppose you are sleeping or watching TV without any worries. Tomorrow you have weekend, okay, so you just have had your food and now you're sitting on your couch and watching TV. So at that time, mainly we say the parasympathetic activity predominates, right? Parasympathetic activity is high in your body at that time. Whereas if you consider another situation, suppose a dog is chasing you, okay? At that time, you will have a condition known as fight or flight, right? Fight or flight condition. Or suppose you are under stress or anxiety. Even suppose you have exam tomorrow. At that time, you are in a big stress, okay? Suppose you are in pain or any other condition. In all those cases, mainly the sympathetic activity predominates activity, right? But we'll be talking about this parasympathetic nervous system today. So, as I told you, parasympathetic activities are also known as cholinergic nervous system, right? So, where basically the word cholinergic came from, as we know, it basically came from the word acetylcholine, right? Acetylcholine, from that cholinergic name. And what is acetylcholine? It is basically a neurotransmitter that plays a role in the cholinergic nervous system. Okay, I'll be talking about that very soon. So, I'm writing a statement which says that the neurons, okay, so the neurons which releases acetylcholine are called cholinergic neurons. Now, so this statement says all the neurons in the nervous system which releases acetylcholine as a neurotransmitter are known as cholinergic neurons. So, a thing might come in our mind that only this parasympathetic nervous system or the cholinergic nervous system will comprise of the cholinergic neurons, right? But that is not true. Well, obviously, the parasympathetic nervous system will contain the cholinergic neurons, but even the sympathetic nervous system also contains the cholinergic neurons. How? Just let me explain you that. So, if you consider CNS, suppose this is the brain and we have the spinal cord. So, okay, let me drop it better. So, this is the brain, this is the spinal cord. Okay, yeah. So, we have the CNS here. Now, all the ANS activity starts from CNS. Okay, so you have to keep this in mind. All the neurons starts, which comes out from the brain only, from the CNS only, right? So suppose some set of neurons are originating from the central nervous system and they are going out to the periphery. Okay, so at some time point they ends up in a certain junction, and these junctions are known as ganglia, right? So some of the neurons which starts from the central nervous system, they goes to the periphery, means outside the central nervous system, and they ends up at certain junction points. Those are known as ganglia. Now, from this ganglia, another set of neurons starts and they reaches to different organs of the body where this ANS is being innervated. For suppose some of the neurons can reach the eyes, okay? Other can go to the suppose lungs or even heart. So like this way, all the organs in our body are innervated by this autonomic nervous system. But the main thing which I want to focus on is this whole set of autonomic nervous system. They are comprises of two set of neurons. This is the first one, this is the second one, and we have ganglia in between them. So this is known as ganglia, right? Now, as per the terminology, you have to keep it in mind that the first set of neurons, which originates from the CNS and it ends up at ganglia, these are known as preganglionic neurons, right? Because as this is the ganglia, and it is present before the ganglia, so it is preganglionic neurons. Similarly, this will be like this is after the ganglia, so it will be postganglionic, right? Now, both of these particular type of neurons contains some a neurotransmitter, because the neurotransmitter release will only cause the signal propagation, and that will innervate the the autonomic nervous system. So these are different from parasympathetic nervous system and sympathetic nervous system. But the thing you have to keep it in mind is the preganglionic neurons in both the parasympathetic and the sympathetic nervous system. These are cholinergic in nature, right? So it doesn't matter if it is parasympathetic or sympathetic, but the preganglionic neurons are cholinergic in nature in both the cases. Well, the neurons are different, obviously, for the cholinergic as well as for the adrenergic. The neurons are different, but they are cholinergic in nature because they release acetylcholine. Now, it has some reasons for this because in ganglia, there are some cholinergic receptors present, which helps in receiving the information from the preganglionic neurons, and they transmit it to the postganglionic neurons. That we'll talk about this later about the nicotinic receptors. But the thing you have to keep in mind is all the preganglionic neurons are cholinergic in nature. And for the postganglionic neurons, now it depends on the type of innervation. Like for the cholinergic, uh, it is it releases acetylcholine, so it is cholinergic nervous system, and or we can say for the parasympathetic, right? And for the sympathetic or adrenergic, it mainly releases norepinephrine. So the postsynaptic neurons, their neurotransmitter differs, but the preganglionic neurons, their neurotransmitter is always acetylcholine. Now, as I told you about all of these things, we will focus on the parasympathetic nervous system innervation in more details. So what basically happens is, let me again draw a brain and a spinal cord. Okay. So for the cholinergic nervous system or the parasympathetic nervous system, usually the neurons which originates from the central nervous system, they starts from two regions of the central nervous system. One is the cranial portion and one is the sacral portion. Okay. So or in other terms, we can also say that the cholinergic nervous system, they are basically craniosacral in origin, right? Because some of them are starting from cranial nerves and some of them are initiating from the sacral region, so they are called sacral nerves. Okay, so basically in this cranial nerves, like we have many cranial nerves, right? But not all of them participate in the parasympathetic nervous system, but some of them, like cranial nerve 3, cranial nerve 7 in the pons region, 9, 10 in the brainstem region. So all these four cranial nerves, they participate in the parasympathetic nervous system. And in terms of sacral nerves, we have 2, 3, and these are S2, S3, and S4. So these are the seven set of neurons which mainly transmits all the parasympathetic activity in the human body. Okay, so basically, if we talk about this cranial nerve 3, so what is cranial nerve 3? It is known as oculomotor nerve, right? So they basically initiate from the cranial nerve 3, and they initially goes to a certain type of ganglia, right? So this is the preganglionic neuron which originates from the third cranial nerve, and it reaches a ganglia. And now this ganglia has certain name, as well as the origin of the third cranial nerve. This particular region has certain name. Okay. Now the region where it propagates, where it initiates and it goes to the periphery, these regions are known as nucleus. Okay, so the third cranial nerve, it starts from a particular type of nucleus, which we know, which is known as. Let me write it here. Edinger-Westphal nucleus, right? Edinger Westphal nucleus or nuclei. Okay, and it terminates at a particular ganglia, and this ganglia is known as ciliary ganglia, right? And as we know, the third cranial nerve. What is the third cranial nerve? It is the oculomotor, right? So the third cranial nerve, the oculomotor, it originates from the Edinger-Westphal nucleus in the brain, and it goes to the periphery and terminates at a ganglia, and this ganglia is known as the ciliary ganglia, right? Now coming to the next one, the seventh cranial nerve. So uh, any idea about what is the seventh cranial nerve? So the seventh cranial nerve is the facial nerve, right? We call it a facial nerve. It starts from a region in the pons, right, in the brainstem, and it ends at another type of ganglia. Okay, so what is the name of the ganglia? The name of the ganglia, like it basically goes to two type of ganglia, like it from a branch. Okay, so okay, let me erase this thing. So basically, it originates like this, and it branched out into two portions, and it forms two type of ganglia. Okay, the first ganglia is known as sphenopalatine ganglia, right? Sphenopalatine ganglia, and the next one is the submandibular ganglia. Submandibular ganglia, right? And the seventh cranial nerve. What is the nucleus known as? This nucleus is called. What should I write here? Okay. This nucleus is known as superior salivary nucleus. As it is being suggested by the name salivary nucleus, so it might innervate to the salivary glands. Okay, so we will talk about innervation later. Right now, we are just talking about the preganglionic neurons and the ganglia names. Okay, and obviously, what is the name of this ganglia? The name of the ganglia is facial nerve, right? So the third one was oculomotor. Seventh one is facial. Third one, cranial nerve 9. Very popular cranial nerve. Okay, well, I would say second most popular cranial nerve, because the most popular one is coming at the end, right? So the ninth cranial nerve, this ends at a particular ganglia, and it is known as oticanglia, right? These are ganglia, right? So the ninth cranial nerve, it ends at otic ganglia, and the name of the nucleus here is inferior salivary gland, right? It also innervates salivary glands, right? So it's inferior salivary nucleus. And what's the name of this ninth cranial nerve? The name of this cranial nerve is called glossopharyngeal, right? Okay, so we are done with this three type of cranial nerves. Now, as I was talking about the most famous cranial nerve, that is the cranial nerve 10, right? So this cranial nerve 10, it again originates from a certain type of nucleus, and it ends at a certain type of ganglia, and this ganglia is known as dorsal motor nucleus of the vagus, or we can say it as DMV. So it is dorsal motor nucleus of the vagus, okay? And this innervates many organs of the body. For example, it can originates from, okay, so it can right from, suppose we have the lungs, right? We have heart, we have the GI tract. If you consider a GI tract here, right from the esophagus, stomach, duodenum, okay, small intestine, large intestine, and not all portion of the large intestine, but till the transverse colon. Okay, so it mainly propagates to the esophagus, stomach, duodenum, small intestine, and some part of the large intestine, like till transverse colon. So the neuron which innervates all these organs are known as vagus nerve. Okay, very important nerve. And the nucleus from where the vagus nerve originates, this is known as dorsal nucleus of vagus. Okay, so this 10th cranial nerve, it originates from dorsal nucleus of vagus, right? So till now we talked about the third, seventh, ninth, and 10th cranial nerve. Okay, their pre ganglionic neurons, what are the name and what are the ganglia name, and we talked about which organs does the vagus nerve innervates. We are left with the innervations of the other three cranial nerves. So basically, what happens, as the name suggests, for the third cranial nerve, the oculomotor nerve, it mainly innervates the eyeball. Okay, so the postganglionic neuron starts from the ciliary ganglia and it goes to the eyeball, right? Mainly the ciliaris, and it also innervates the pupil. Okay. Now, the seventh cranial nerve, as it branches into two parts, right? So the sphenopalatine, it goes to the mucosal gland, mainly of nose, pharynx, and buccal cavity. Sorry. And the submandibular ganglia, this mainly innervates some of the salivary glands. For example, submandibular and sublingual. And the last one, the otic ganglia, it mainly innervates the parotid gland, which is also a type of salivary gland, right? So we got all the innervation of the cranial nerves, which are a part of cholinergic outflow. Now, once we are done with the cranial innervation, next we have the sacral innervation. Okay, so as I said, some of the cholinergic neurons, they originate from the sacral portion of the central nervous system, mainly from the S2, S3, and S4 regions. So they also starts. Okay, so they also originates from the spinal cord sacral 2, 3, 4 region, and they ends up, the preganglionic neuron ends up at certain ganglia, and then further the reaches to the lower part of our body, especially after the transverse colon, rectum, and some genital organs as well. Okay, so okay, let me draw it here. Okay, so basically, if we consider the sacral innervation, some of the innervation goes to the suppose the distal colon, some of them goes to rectum, anal sphincter, some of them can also goes to the genitalia, right? So by this way, most of the organs of our body are being innervated by the cholinergic nerves, and this is very important for the proper function, right? Now, one of the thing you have to keep it in mind, like as I told you about the preganglionic neurons and the postganglionic neurons. So what happens in case of parasympathetic nervous system is the preganglionic neuron is very long. Okay, so for parasympathetic neurons, okay, so let me again draw brain and spinal cord. Okay, so suppose the cranial nerves are originating from this region uh, of the central nervous system, and so the preganglionic neurons will travel a very long distance near the organ. Okay, and they will end up in the ganglia. So the preganglionic neurons, these are very long, whereas the postganglionic neurons are relatively short because these ganglia are present near the organs. Okay, so the postganglionic neurons will be short. But this thing is completely opposite in case of sympathetic nervous system, as if I again draw brain and spinal cord, suppose the neurons which are originating from the central nervous system are very short, and that the reason is because the ganglia are present just near the central nervous system for the sympathetic nerves. Okay, okay, so let me draw it in a better way. So suppose the ganglia are present just near the central nervous system, so the preganglionic neurons don't have to travel a long distance. Okay, so they are relatively shorter, but the postganglionic neurons are quite long. Okay, so they have to travel a long distance in order to reach different organs. So this is one of the fundamental aspect you have to keep it in mind about the difference between parasympathetic and sympathetic nervous system. Okay, so the next part is we have to know about some more about acetylcholine, right? So let's talk a bit more about acetylcholine. We have to know some of the aspects of this particular neurotransmitter, like their synthesis, storage, their release, their effects, and the receptors which they activates. Okay, so these are the fundamental aspects that you have to know about this particular neurotransmitter. So as the name suggests, acetylcholine, this is not a peptide hormone or a neurotransmitter. Obviously it's not a peptide. Like many students confuse it with a peptide. Okay, so as it is not a peptide, so suppose a neuron. Okay, so this is just a rough diagram of a neuron. So as it is not a peptide, so this acetylcholine will not be produced in the cell body or the neuronal body, right? Because if it is a neuron, then only the it has to undergo transcription, translation, and then it will come out of the nucleus, and then it will form a protein, right? But as it is not a peptide, so it will not produce in the cell body. Rather, it will produce from that particular region where it will function, or at the region where it will release to cause some effect. So mainly it is being produced at the nerve terminal. Okay. So all the small molecules like acetylcholine or any other neurotransmitter which are not peptide in nature, they are generally produced at the nerve terminal. They are stored at the nerve terminal so that it will be easier for them to release whenever required, right? So one of the properties of the neurotransmitter that are stored in nerve terminal, they are basically small molecules, right? And they are non-peptide. Because if they are peptide, they have to be, transcripted, translated, and for that we need nucleus, right? As nucleus are not involved in this non-peptide molecules, so they are usually synthesized here and stored here only. Because if they are synthesized in the cell body, then it will take ages for them to come into the nerve terminal in order to release. Okay, so that's one thing. It is not only limited to acetylcholine. For example, some other neurotransmitters as well. If you consider GABA, GABA. Yeah. So this is another neurotransmitter that is also non-peptide and it is usually stored in a nerve terminal. Some other examples like glycine, glutamate. Okay. So, uh, so the main thing is that acetylcholine, as it is synthesized and stored in the nerve terminal, now we have to see how acetylcholine is synthesized in the nerve terminal. So if you zoom out at this particular nerve terminal, okay. So suppose this is a particular nerve terminal, okay? And first of all, we have to know certain things that uh, so acetylcholine is usually formed by two molecules, okay? So that is acetyl coenzyme A plus choline that makes acetylcholine, right? Now, so we have two components here. One is the choline and another is the acetyl coenzyme A. Now, among these two, between these two molecules, one of them is being taken up from the external sources, mainly from the diet, and one is produced in our body. Okay, so between these two molecules, usually choline is taken from the external sources, for example, from the diet, but acetyl coenzyme A, it is formed in our body, right? So first, if you have to talk about how choline is being transported from the diet to the nerve terminal, so choline is generally taken up from different sources of food, and then it goes into our circulation, and some of the choline moves from the circulation to the extracellular fluid, and from the extracellular fluid, they are taken up by the nerve terminal. Okay, so for that you have to know some of the sources of choline. Okay, it might be helpful for your day-to-day life as well, you know. So, some of the foods which are very rich in choline, this includes the most important one is the meat, right? Meat, fish. Now, if you consider any vegetarian options, then obviously soybean, potatoes, broccoli, and some nuts as well. For example, peanuts. So, and many other foods are also there. So these are the important sources of choline. So when we take this particular type of meals, choline is being uh, taken into the circulation, and from the circulation, so from the diet, it goes to circulation. From the circulation, it goes to the extracellular fluid, and from the extracellular fluid, it goes to the nerve terminus. Now, how choline goes from the extracellular fluid to the nerve terminus? Suppose this whole thing is the extracellular fluid, right? Now it has to go to the nerve terminal. Like it cannot directly pass uh, the nuclear terminal membrane, right? So there has to be some certain gate for the choline to get enter, right? And that gate is usually called CHT. So it's basically a choline transporter. Okay. So suppose this is a gate, and this is known as clean some space here. Okay. So this is basically known as high affinity choline transporter, or it is also called as CHT1. Okay, so what it does, it basically helps for the intake of choline into the nerve terminal, so the choline can come inside, right? And it is basically a type of symporter. So it causes influx of choline, as well as it causes influx of one molecule of sodium. So you have to keep it in mind that the CHT1 transporters, they usually act as a symporter. They causes the influx of choline molecule from the extracellular fluid, as well as one molecule of sodium ion. Okay, so now we know how choline is formed in how choline is being taken up by the nerve terminal, right? Now the next thing is, as I told you, so acetylcholine is formed by acetyl coenzyme A and choline. Now we know about choline. We are left about acetyl coenzyme A, right? So you might have studied in biochemistry about glycolysis, Krebs cycle, or TCA cycle, right? So in that particular cycle, generally acetyl coenzyme A is formed by the changes in the pyruvate. Okay, so I will talk that a bit more. Just as a recapitulation, so that you can remember how acetyl coenzyme A is formed via glycolysis. So, how acetyl coenzyme A is synthesized, right? All right. So, suppose we are talking about glycolysis, obviously from glucose. After the end of the glycolysis, we get two molecules of pyruvate, right? Along with two molecules of pyruvate, obviously we get energy because that the main motto is to get energy from carbohydrate sources, right? So we get two ATP and we get two NADH molecules, right? So now this pyruvate has to recycle. So this pyruvate, and we know that this particular thing occurs in cytosol, right? So this pyruvate has to go and join into any other type of pathway. So it enters the mitochondria of the cell. Okay, suppose this is the mitochondria. So there is a specific type of ion channel, not ion channel, like a transporter present on the membrane of mitochondria that helps in the intake of pyruvate into the mitochondria. And this particular transporter is known as mitochondrial pyruvate carrier. Okay, or we can say MPC, right? So it's a bit easy name. Mitochondria, as this membrane, this is present in the mitochondrial membrane. Mitochondrial, and it is carrying pyruvate molecule, so it is been named as mitochondrial pyruvate carrier, right? Like no fancy name, just simple. What work it does, that's how it is named, right? So once it enters the mitochondria, now inside mitochondria, so we have uh, apologies. Yeah. So inside mitochondria, we have pyruvate. And from the uh, mitochondrial respiration, we get coenzyme A and NAD plus. All right. So we have these three molecules. Now these molecules interact with each other to form acetyl coenzyme A plus a molecule of carbon dioxide and one uh, NADH. Right. So what basically happens is pyruvate as like if you see the structure of pyruvate, it is having three carbon molecules. Okay. Now this three carbon molecules and there is one coenzyme A present is being converted into acetyl coenzyme A and one carbon dioxide. So acetyl coenzyme is two carbon and this is one carbon. So basically the pyruvate molecule breaks down into a two carbon molecule and into a one carbon molecule, right? So if you see, it basically has acetyl and it also breaks into a carbon dioxide group, right? CO2. And acetyl is, we know, acetyl group, this one, right? So finally, we get acetyl coenzyme A, that is CoA and carbon dioxide molecule, right? So now this acetyl coenzyme is formed in mitochondria, right? But acetylcholine formation takes place in cytosol only. So this particular acetyl coenzyme A has to come into the cytosol from the mitochondria again, right? And acetyl coenzyme A cannot come or cannot cross the mitochondrial membrane of its own. So it has to be modified in order to cross the mitochondrial membrane. So what happens? This acetyl coenzyme A, this usually interacts with another molecule that is oxaloacetate. Okay, so once it interacts with oxaloacetate, it usually forms citrate. Okay, so this is a very important molecule as it helps in the transport of this acetyl coenzyme and oxaloacetate from the mitochondria to the cytoplasm. Okay, citrate can easily pass or cross the mitochondrial membrane and so cross the mitochondrial membrane, right? So once it crosses the mitochondrial membrane, in the cytosol, it again transforms into acetyl coenzyme A and oxaloacetate. So we get free acetyl coenzyme A that can be helped for the synthesis of acetylcholine. Okay. So what really happens? This citrate molecule crosses the mitochondrial membrane. So if I draw a mitochondria again, right? So there is another transporter molecule present. The name of the transporter molecule is citrate transporter, and it helps in the transport of the citrate to the cytosol. Okay, and in the cytosol, this citrate molecule interacts with one ATP to form acetyl coenzyme A and oxaloacetate. And so obviously we need an enzyme in order to form a reaction. So the name of the enzyme is ATP citrate lyase, right? So just as a short recapitulation, what happened is from the glycolytic pathway, as we know, glucose converts into energy source. So we got two ATP molecules as well as two pyruvate molecules. Now this thing happened in cytosol. So this pyruvate then moved to the mitochondria by using via some molecules, carrier proteins, and inside the mitochondria, it interacted with coenzyme A and NAD plus to form acetyl coenzyme A, right? So this acetyl coenzyme A reacted with oxaloacetate to form citrate. Now this citrate can cross the mitochondrial membrane, as acetylcholine cannot cross the mitochondrial membrane, so it has to be converted into another form that can cross the membrane, right? So then it formed citrate, and citrate came back to the cytosol, right? So this is cytosol, and this is mitochondria. Okay, so it came back to the cytosol, and then cytosol reacted with a molecule of ATP and using an enzyme called ATP citrate lyase, it again formed acetyl coenzyme A and oxaloacetate. But we are focused on this acetyl coenzyme A. So by that way, we found uh, acetyl coenzyme A in this particular reaction, right? So okay, so in the nerve terminal, we have the choline and acetyl coenzyme A at the moment, right? Now what happened is both of them react with each other and they form a molecule of acetylcholine. Now, this reaction does not take place of its own. Obviously, it needs some kind of enzyme that will initiate the reaction. So the name of the enzyme is choline acetyl transferase, or CHAT, right? I can write it here. CHONE, choline acetyl transferase. that can form a molecule of acetylcholine. So by this way, acetylcholine is formed in the nerve terminal. Now, this formed acetylcholine has to be stored somewhere in the nerve terminal because they cannot be freely available in the cytosol. They will degrade. They will come in contact with different enzymes, and finally they will break down into acetyl coenzyme again. So they has to be stored somewhere, and they are usually stored in certain vesicles. Okay, so there are certain vesicles formed throughout the nerve terminal in which these acetylcholines are transported and then they are stored in these particular vesicles. Now, one thing you have to keep it in your mind. Now, again, acetylcholine, they cannot just directly go into the vesicles. There has to be some transporter molecule which causes the entry of acetylcholine in these vesicles. Before that, these vesicles, these are basically lipidic in nature. So these are lipid vesicles. Okay, so they are generally made up of lipids, and we know lipids are not soluble in the hydrophilic substances present in the cytosol, so they remain as a circular vesicle roaming around the nerve terminal in the cytosol, right? So, what happens is there is a particular transporter molecule again. Okay, so there is a transporter molecule present which causes the entry of acetylcholine into the vesicles, and the name of that is VSACHT, vesicular acetylcholine transporter, right? Vesicular acetylcholine transporter. Now, this is basically a type of channel that causes influx of acetylcholine, but on the other hand, it also causes efflux of a proton molecule. Okay, so it is causing influx of acetylcholine, but at the same time, it is also causing influx of proton molecule. Now, how does this work? Let me explain you this a bit. So basically, what happens? Suppose this is a particular vesicle, right? And we have the transporter present here that causes influx of acetylcholine and efflux of the proton molecule. Right? Now, from where this proton came inside this vesicle? So this came from another type of transporter. Right? Suppose this is another type of transporter, and this transporter is called frequency proton ATPase. So is ATPase, right? So this causes what? This causes influx of a proton molecule inside the vesicle. Okay, and what is the need of this proton molecule? So basically, this VACHT, this requires a proton for its energy for it to transport a molecule of acetylcholine, right? So it transports a proton molecule. Simultaneously, it gets energy. It gets the signal that okay, I have enough proton inside me, and now I have to throw away some proton from my body, and in the exchange of that, I will take up some other molecule, and that molecule is acetylcholine. So basically, it is getting some instruction from the proton that I will throw up that proton and I will take something else, and that something else is known as acetylcholine, right? So by this way, basically, acetylcholine is stored inside these vesicles until they are released. But acetylcholine is not only stored inside these vesicles; they are usually present with some other molecules as well. Okay, so just one thing you have to keep in mind that acetylcholine is basically positive charged molecule. Okay, so these are positively charged, and as also VACHT, these are a type of exchanger. So it exchanges, it throws away a positive molecule and it takes into the vesicle a positive molecule, right? So as positive charge is going out and positive charge is going in, so there is no change in the homeostasis inside these vesicles. So acetylcholine are basically positive charged molecules, and these are usually bounded to some other negatively charged molecules inside the vesicles in order to stay stable inside the vesicles. Okay, and these are usually present or bound to ATP molecules. Now, ATP are negatively charged because of the presence of phosphate, right? So these are present with the ATP molecule, which are negatively charged, and acetylcholine are positively charged. So positive, negative, they usually don't exist. So they combines together and they remain as an uncharged molecule, right? And some other glycoproteins are also present apart from acetylcholine and ATP. Some we can find some other glycoproteins present in this particular vesicles. Okay. So this thing, I think this is a bit important to know that it's not only acetylcholine, but apart from acetylcholine, there are some ATP molecules, and obviously there are some glycoproteins present inside these vesicles. Okay, so the question can arise is why acetylcholine is stored in the vesicles, right? Like I have seen in many examinations, this type of questions are asked: why acetylcholine is stored inside vesicles? Now, I have answered you this previously, just again to write it in the note that the most important thing, the cytosol contains a lot of acetylcholinesterase. So if acetylcholine is present in the cytosol for a very long time, that cholinesterase molecule will break down the acetylcholine, right? So it will again form acetyl coenzyme A and choline. So we don't need that. So in order to prevent degradation in cytoplasm by different enzymes, that's why uh, they has to be transported into the vesicles. Okay, and another thing is that as the vesicles are mobile, right? So as the vesicles are mobile, during the time of exocytosis, these vesicles can easily come near the nerve terminal membrane, and they can cause exocytosis to release acetylcholine to the synapse, right? So during the time of exocytosis, the vesicles can reach the nerve terminal and released quickly, right? So these are one of the like two of the important factors why acetylcholine is stored inside the vesicles. And another thing, like just for your knowledge, I would like to say that each vesicles contains around 5,000 to 10,000 acetylcholine molecules, right? So this is the average number of acetylcholine molecules present in each vesicles, and there are like thousands of vesicles in a single nerve terminal. Right now, there is a concept of physical organization that you might not find in many textbooks or in many places. So this is relatively a new term that scientists have discovered, that is a physical pool organization. What happens is, so these vesicles are present like an organization. Okay, so suppose I'm drawing a nerve terminal. So this is suppose a nerve terminal. Now inside them, some of the vesicles are suppose present near the nuclear membrane where the exocytosis takes place. Okay, some of them may be present in between the nerve terminal body. Some of them can be present at the extreme deep of the nerve terminal. Okay, so they are recruited like an army. So some of them are present near the place where exocytosis will take place, like this. Some of them might be present at the middle portion. Some of them are present at the deep in the nerve terminal. So they're recruited based on the order they're placed. Okay, so these these vesicles, these are readily releasable, right? And we call it as a pool. So they are present in one pool, they are present in second, and they are present in the third pool. So these vesicles can release or exocytose very quickly upon their stimulation, right? The second layer is the recycling pool. And the third layer is the deep cytosolic, or we can say it as a reserve pool, right? Okay, so this is one of the important aspects that you have to know about the organization of vesicles in the nerve terminal. Okay, so now we know about the synthesis, storage, and orientation of acetylcholine in the nerve terminal. So some of the other things are left before going to the release of acetylcholine. Uh, so we have to know about at what particular locations acetylcholine acts as a prominent neurotransmitter. Okay, so we can write it as acetylcholine is a main neurotransmitter at, so there are certain regions, especially as we all of us know, this has to be cholinergic neurons, right? So uh, parasympathetic, preganglionic, right, neurons that we discussed before, that the preganglionic neurons, let it be parasympathetic, let it be sympathetic, both are cholinergic in nature. So obviously sympathetic preganglionic neurons. Okay. Apart from that, there are some nicotinic receptors present in the ganglia. Okay, so we can write it as autonomic ganglia. There are some nicotinic receptors also present in the neuromuscular junction for the somatic nervous system. So this doesn't becomes a part of autonomic nervous system, but still these are cholinergic in nature, and they mainly contains NM type of nicotinic receptors. So we can write neuromuscular junction, although they form a somatic nervous system part because they involve skeletal muscle. Okay, and at last, many CNS synapse also contains uh, acetylcholine. Okay, and you have to keep it in mind that uh, this acetylcholine plays a very important role in cognition and memory, right? That's why people who have Alzheimer's, people who are suffering from Alzheimer's disease, they usually have imbalance in the acetylcholine level, and that causes them to, lose memory. So now one question arises is mainly the acetylcholine is formed in the nerve terminal, right? Or maybe this choline that we discussed earlier, the choline molecule which forms a part of acetylcholine, this choline molecule is being transported from the diet, right? From the extracellular sources. Then how it is entering the brain to form acetylcholine? Because in brain, as I told you, acetylcholine plays a very important role. So how it enters or crosses the blood-brain barrier to form acetylcholine and function in the CNS. One important thing about this is acetylcholine usually acetylcholine is usually formed inside the CNS, right? Now, how choline is transported? Choline is usually transported via formation of phosphatidylcholine. Okay, so choline forms phosphatidylcholine and it then enters the central nervous system. Okay, it crosses the blood-brain barrier, it enters the central nervous system, and then it interacts with acetyl coenzyme A to form acetylcholine. So, an acetyl coenzyme is obviously formed in the CNS only. So, this is the way by which acetylcholine is also formed inside the central nervous system. Okay, so finally, we have to know about the release of acetylcholine from the nerve terminal. Release of acetylcholine, right? So, we have to see how the acetylcholine, which is present inside the vesicle in the nerve terminal, are exocytosed so that the acetylcholine can be released into the synapse and then it can bind to its particular receptors. So, let me draw a big nerve ending again, right? Just for recapitulation, as we talked about before, about choline. So choline enters the nerve terminal using a receptor or a transporter known as CHT1, right? So choline entered the nerve terminus. It reacted with acetyl coenzyme A, right? Acetyl coenzyme A, we get it from glycolysis pathway. From the mitochondria, it comes to the cytosol, right? And then it forms uh, acetylcholine, right? So we got acetylcholine by this way, and obviously it involves an enzyme CHAT, right? Choline acetyl transferase. Uh, so right now we know that this acetylcholine, once they form in the cytosol, they are transported to the vesicles, right? So the vesicle, they are transported into the vesicle using some proton exchangers like VACHT. Okay, these are some transporters. These are also called as proton exchangers. Okay, so now whenever required upon stimulation, this acetylcholine has to be released into the external synapse. Suppose this is the synapse, and we have a certain kind of say smooth muscle or any other maybe skeletal muscle or maybe another neuron present on the other side, okay, of the other side of the synapse. Another side of the synapse uh, may have some of the receptors. It could be G protein receptor, it could be ion channels that we will talk about this later in the classifications of the cholinergic receptors, right? So, how this acetylcholine can be released into the synapse, that's the important thing. So, to know that, we have to focus on this particular vesicle. Okay, so these vesicles are not just a lipid bound membrane. They contains a lot of things. They process a lot of things. As I told you before, this vesicle contains not only acetylcholine but also ATP and some other glycoproteins as well, right? They have some different type of transporters like proton transporter and proton ATPases as well. So these vesicles play a very important role in the nerve terminal. Okay, so as I was talking about like how the exocytosis of this acetylcholine can take place. This usually happens via the utilization of some proteins. So there are some proteins present on the surface of the vesicles. Usually, there are two types of proteins present. One is, as you can see, just for a diagrammatic depiction, I am making a straight line. Okay, so this is one type of protein, and this is known as synapto synaptobrevin, right? So this protein is known as synaptobrevin, and we have another type of protein present. Suppose it is like this, it is closed. Okay, suppose this is the active chain and this is closed, and this protein is known as synaptotagmin. So just keep it in mind that this vesicle contains two type of protein on their surface. One is the synaptobrevin and another is the synaptotagmin. Okay, and now this synaptotagmin's head portion is usually in a conformation which is closed, right? So nothing can bind to this at this point. So this is a closed position, and other protein is the synaptobrevin. Now what happens? There are some other type of proteins also present uh, on the surface of the nerve terminal, right? So this is also a lipid bilayer, right? And there are certain proteins present on the surface of the nerve terminal, on the membrane of the nerve terminal, in better terms, we can say that. And these proteins are called syntaxin or SNAP25, right? So these proteins are syntaxin and SNAP25, and the proteins in the vesicle are synaptobrevin and synaptotagmin. Okay, so what really happens is usually at the resting stage, the potential, the membrane potential or the resting membrane potential is very negative, right? It is very negative. But what happens if under stimulus, if by somehow it got the stimulus and depolarization is taking place? In that case, as you know, the nerve contains a lot of voltage gated sodium channels, right? So they have a lot of voltage gated sodium channels, and they are fast inactivating channels, and usually they cause influx of sodium ion upon change in the voltage. So what will happen whenever depolarization will take place, whenever there is some stimulus? In that case, the sodium influx will take place inside the cells, right? So uh, so these are the voltage gated sodium channels. And when the sodium influx takes place inside the cells, this further activate another type of ion channel that is called LTCC or L-type calcium channel. Okay, so these L-type calcium channels are also called as voltage gated calcium channels. Sorry, I think I made some mistake. It won't be L-type calcium channel. Oopsie. Yeah, so it won't be L-type calcium channel. It would be either P-type or N-type calcium channel, right? L-type channels are present mainly in the smooth muscle cells. Okay, yeah, sorry, my bad. But these are also voltage gated calcium channels, okay, and they causes huge influx of calcium from extracellular region to inside the cell. Okay, so as these are voltage gated, so if there is too much sodium coming inside the cell, see, in the resting stage, the cell is in negative potentials, right? But under the depolarization, under the stimulus, too much sodium is coming inside the cell, and we know that the sodium is positively charged ions, right? So from negatively charged, the potential will start to rise slowly until it reaches the threshold potential where these calcium channel voltage gated calcium channels are activated. And once these calcium channels gets that threshold, it will cause huge influx of calcium in it, and we'll see a depolarization or a contraction, right? So once we see a contraction, at that time, so many calcium will be inside the cell. And this calcium plays a very important role in the exocytosis of the acetylcholine. So what happens is when this calcium is inside the cell, they go and bind to this particular position, which was closed before. Okay. So once they bind there, what happens? Now the closed conformation will be changed to open conformation. So suppose this is the acetylcholine molecule. Okay, let me draw it here. Suppose this is the vesicle and it has acetylcholine inside it, right? Uh, yeah. So earlier the synaptotagmin that was present, now it will grab the calcium molecules in it. Okay. So the calcium will come and bind to it. Once this calcium comes and binds to the synaptotagmin molecule, it goes near the syntaxin, okay, and it forms a bond with syntaxin. Okay, it formed a bond. Similarly, we had synaptobrevin as well attached to the vesicle. That vesicle will also changes its conformation and it also binds with the syntaxin or the SNAP25. Okay, so it is like this. Both the proteins will bind to the syntaxin and ultimately both the proteins will push towards the syntaxin. So it will typically exert a push-like motion in this towards the nuclear, I mean the nerve terminal membrane, right? And ultimately, what will happen as this push is too much, that will cause the fusion of this lipid membrane vesicle to the nerve terminal membrane. Okay, so what usually happens is if the push is too much, the vesicle membrane get fused to the nerve terminal membrane, and all the proteins are obviously present just like this only. Proteins are present like this. It just fused, the two lipid membrane just fused together. And that will cause exocytosis of the acetylcholine molecules because, okay, so it will cause exocytosis of the acetylcholine molecule because, lipid membrane, they fuse together, they will form an intact membrane like this, okay? And whatever present inside the vesicle will come out from the vesicle. So it created a roadmap for the acetylcholine to leave the cell and present in the synapse. And now this acetylcholine can bind with its muscarinic or nicotinic receptors and then exert its secondary messenger pathway to show its effect. So it could be M2 receptor, M3 receptor, NN receptor, NM receptor. Okay. By this way, it further exerts some effect. We'll talk about the effects later, but I hope you got the basic phenomena, what happens, uh, how the acetylcholine that is formed inside the vesicle is released from the nerve terminal and how it works, how it is stored, how it is made, and the basic theory about the parasympathetic nervous system. I hope it is clear to you. If you want to learn more, if you have any doubt, you can always comment and I will try to answer all of you. And have a nice day. We'll see you in the second part.